Hi. This is Miss Litton, and this is 7th period. I have some wonderful AP biologists here. Say hi. Hi. Okay, and we are reviewing Unit 5, and I just wanted to bring up the list of all the topics. And the reason why Tristan, we have been waiting and waiting for you. We thought you were coming. Okay, and so, and the reason why I'm bringing this up is any of these things, even though I had set out some token essays that it could be, okay, really the essays can come from any one of these topics. Where would be some ways for you to find some other potential essays than the ones I already listed for you? Where could you look? In the back of each chapter, right? And they have science practices you could look at. You could look at any of the thinking questions at the back and consider those as viable options, you know, to prepare for your AP exam. So viruses, bacteria, and archaea, I know I have at least one question on viruses. I know I do. But then, hi, sweetheart. And then bacteria and archaea, you could think of ones even on your own. How are they similar? How are they different? Could you see like a Venn diagram? Where I had double circles, and I had that out on for your question, and I had both, and then I had bacteria and archaea, and I had you list things inside a Venn diagram. Wouldn't that be a good way for me to tell whether you knew the differences or not? That's really good. I should do that. Okay? Chapter 23, 24, plant evolution and structure. You know we jumped around. There were only certain things you needed to know, but things you want to walk away with. When we talked about plant evolution to get on land, that was a big deal, right? So knowing the steps of that, stuff like protecting the embryo. Yay. You know, getting that water, having xylem and phloem, right? Having a seed, having a flower. All of those items, those are good things. So just kind of look over at the big ticket items and go, hey, I could write about that. Structure. What are some structure things maybe I didn't ask you a question about on the essay that you might want to know the differences between? Like monocots and unicots. That might be something I could ask you about. Identify 4 monocot traits, 2 eudicote traits, or something like that. That would be a good question. Flowering plants, nutrition and transport. Okay, so what's nutrition about? What's plant nutrition about? How they, what? What are the things plants have to acquire? Let's talk about that first. What are things plants have to acquire? Water. Listen to this. He walks in late and he just like boom it. Water, carbon dioxide, what else? Remember C. Hopkins Cafe, cousin mine, managed by my cousin Kleinbo. So they need to get things out of the soil. Do you remember the important ones like nitrogen needs to come out, potassium, phosphorus? In fact, if you look at any fertilizer, it'll always give you the NPK percentages. Nitrogen, phosphorus, potassium, because it may be dependent on that particular organism. So you need things from the soil. Do plants need oxygen? Yes, for what process? Cellular respiration. And that's usually not an issue in your photosynthetic parts of the plant, right? Because they're generating a lot of oxygen as a result of photosynthesis. And in fact, it can be a problem how much oxygen they're generating because you're fearing what? Yeah, what is that thing? Come on, bring it. Photorespiration. And we know how certain plants have evolved mechanisms in order to avoid photorespiration, like what kind of plants? C4, right? C4, and they use PEP and the bundle sheath cells with the Krantz anatomy. Do you see how I'm talking about this unit, but yet we're finding connections to other units? Remember on the AP exam, there are no units. Okay? No units, it's just science. So don't think of your topics. The more you know, the more you ought to be able to make those connections to those other units. And when you want to support your points, don't put yourself in a box. Nobody puts baby in a corner, okay? Don't put yourself, movie, anyone? You know what movie that is? It's about dancing, naughty dancing. There you go. All right. So don't put yourself in a box with your responses, because you can pull from anything that we have talked about to support and to give evidence for whatever claim you make in an essay. Don't limit yourself anymore. We're now in second semester, and you have a wealth of information to support yourself with. Okay? So you could talk, there was C4 plants, and what was the other one? CAM plants. When they fix CO2 at night, remember that? Oh, yeah. Okay, so anyway, oxygen is usually not a problem. Usually they're trying to figure out ways to prevent photorespiration from happening. But we did see issues with oxygen in roots, right? Because if the soil is too fine of particles like clay, then it doesn't create any space, and so they have a hard time accessing oxygen. And we looked at those roots that would come down and back around in order to get the oxygen they need for cellular respiration. So when we talk about nutrients that a plant needs, okay, yes, they need CO2. Yes, they need nitrogen. Yes, they need items from the soil. Yes, the roots, if they're going to stay alive, they're still going to need to have some access to oxygen. But how do plants generally get carbon, the carbon they need? Through what? Stomata. Okay, carbon dioxide. So you could talk about that. Okay, how do they generally get the water they need? Through the roots. Okay, how do they prevent too much water loss? Closing their stomata. But can they completely close their stomata all the time? Why do you even have stomata? To get gas. You got to have openings to get gas exchange, but you don't want to lose too much water, right? And then when you talk about nutrition and transport, well, if they need water in the roots, how do you get the water to go in the roots? What do? You make those roots hypertonic. That's right, buddy, you do. Okay, so you could use something like a pump. That's right, pumps hydrogen out. He can escort in negative ions or exchange for positive ions. You can concentrate your minerals inside your root and get water to follow it in. Especially if you get it in past what? Where's an interior barrier that's very helpful inside the root? Endodermis. Okay? Those endodermal cells around the stele, around the xylem and the phloem, they have what going around their outside? Casparian strips, which forces water to go through the symplast, where it can be more regulated than going between using the apoplast. Good. So now our water is going in. In fact, if you have it concentrated enough inside the roots, water will keep going in, and the only place then it can go is up. And we call that something, yes, say it.
Root pressure.
Root pressure. And there's evidence of root pressure when a cut stump bleeds or when it gets pushed out the ends of leaves, gutation. Remember that? That's evidence of root pressure. A cut stump or when its water is like squirting out the ends of the leaves. Okay? But root pressure will get your party started, but it's not enough to get it up to the very top of the tree, right? Then we enlist what? TATC theory, yeah, right? Okay, so that is the overview of 25. I'm still going to visit your essays. I'm just kind of hitting all the big ticket items in each one of these. In 26, flowering plants, control of growth responses. So what are some ways to control growth responses we can use? What's the first thing we talked about? Oh, that's great. That is a what though? Hormone. So hormones, right? We can use hormones to control growth responses. And we talked about the 5 different hormones, but the really one you need to know is auxins, because it's involved in so many things. That would be a really good essay question. You know, what critical hormone group do all plants need? And, tell me 4 different items it controls. Why is it important? Okay, and then you can go through those. So we can have control of growth responses using auxin. What's another one? Gibberellin, our party hormone. Gibberellin is like auxin in that what? Yeah, you can still get elongation. Another one that makes things bigger but not through elongation is cytokinin. Cytokinin likes to work with auxin in the shoots and the roots, and it does that by making more what? Cells. Good. What else does gibberellin do?
Or
No, not
the other thing.
Yeah,
Yeah.
it's party hormone. It's saying break out of the dorm, right? Okay, so it breaks seed and bud dormancy. Breaks seed and bud dormancy. And then, so we said auxin, gibberellin, cytokinin. Let's talk about, now we've made that baby because our flower's out there, right? Made that baby. And now it's surrounded by what? Fruit, and it ripens and it drops. Who does that? Ethylene, yes. And then it's getting cold now. Our fruit is dropped. We're moving into winter. We're going to talk about APBio, abscisic acid, getting ready for winter. But we don't just talk about abscisic acid in the winter. We can talk about it daily when it closes our stomata, right? Okay, so those are 5 different hormones that play, groups of hormones that play critical roles in regulating, right here, our responses. Now, we talked about other things. We talked about just the idea of tropisms, where you're growing where?
Towards or away.
Yeah, towards or away from a stimulus. So one of the stimuluses that we would be concerned about is what? Light. So the idea would be I want to grow towards the light, so I expose more of my leaves to that light. How do I accomplish that? Well, it comes from what? Auxin moving to the shady side, right? And then the shady side elongates and tips the rest of the plant to the sunny side, right? And that's how it does that. Now, this is really a good example of a signal transduction pathway. So you receive this signal, like auxin, and it does a couple of different things. What does it do? It goes out into the what? It comes in the plant, or back up. Stop, Whitney. Okay, so auxin binds to a what? Receptor. This triggers in the interior a series of reactions, which eventually gets what to go out? Hydrogen to go out into the cell wall, which lowers the pH and makes it weak. So when water goes in the vacuole, it expands it. And then it also triggers in that signal transduction pathway DNA, and it's eventually going to make more cell wall materials, right? Okay, good. All right, so those are phototropism. What's another tropism? Gravitropism, okay? So this is when plants generally, the roots are going to grow down and the shoots are going to go up, okay? So in gravitropism in roots, it's a little bit different because the auxin goes to the lower side and it causes the cells on the other side are the ones that end up elongating and it turns the roots down. What's another tropism? Thigmotropism. What's an example of thigmotropism? Responding to touch or an object touch, an insect, and it's localized movement. Usually very rapid, if it's like catching a bug or you're curving around something in order to get to a higher elevation to expose more of your leaves to light, right? What's the whole plant response? Stigmomorphogenesis, yeah. Trees on the edge of the windy cliff, and they thickened in order to support themselves against it. What are some other movements? Very spastic, nastic movements are those quick movements like leaves closing when bugs are on them. Still a touch thing, okay? Good. So tropism is a response. Now, another thing we talked about was plants can also respond by going towards the light or closing their leaves, but plants can respond by generating flowers too. And to know when to flower and when not to flower, they have to be able to tell the time. So what do we use to tell time? Phytochrome, exactly. This is like a toggle switch. PR, right? And then you absorb what kind of light? Red light throughout the day, and then it becomes far red light into the evenings, right? And at that point, hello.
Hello.
Do you need me?
No, I was pleased to know that the new swimming pool is coming in here.
I was unaware of that, but that is good to know.
With a diving board.
Excellent.
We help.
Thank you. Okay then. All right. Okay, so PR becomes PFR towards the evening, and then at night, when there's no light, it kind of slowly becomes what again? PR. So it's changing its shape throughout the day. It's like a toggle switch. And then we talked about that when you look at plants, some plants are classified as long day, which really means short night, and ones that are short day, which really means long night, right? And then there are some that are day neutral. And so they have, if you are a long day plant, you with me? That means you need a, so do you have a max or a min number of hours? Max. You need a short night. So you max out. If you go over that maximum, you're not flowering. Okay? Whereas if you are a, what do I need to say now? Short day, and you need a long night, you need a minimum number of hours before you flower. Okay? And then you can use that PR, PFR ratios for other things too. If you're not getting enough PR, maybe you need to get yourself in a better position by growing what? Up. And so you will then focus your resources on growing up, and so that you can cast the shade on the other plants, right? Maybe you change the pH of the soil a little bit and mess them up a little bit. Maybe you're in the dark. If you're in the dark, you do not want to focus on being green. You want to focus all your efforts on growing what? Tall. Yeah, up. And so you'll become green once you come in contact with light. And those are all things we can see. Now, another thing that you can respond to is you can respond to intrinsic signals, you know, hormones telling you what to do, but you can respond extrinsic things that are coming at you. It's not like you're just sitting out there, whatever. You could protect yourself from extrinsic things. What are some ways you can protect yourself from extrinsic things? You could have a symbiotic relationship. Good. You could have, let's start with the simplest, where it's just all by your onesie. You could have what around the outside of you? Rough. Thorns. What else? Bark. Exactly. So you can have protective layers on the outside so things don't bother you. Thorns, spines, you could have toxins, poisons, right? That just protect you. But you could also elicit help through that what kind of relationship? Mutualistic relationship, where you say, I'm going to produce a food for you. You come over here and eat all the bugs that are starting to eat me, and I'll feed you specifically. Or I will create a home for you, little, hollow thorns for you to live in, and then you can attack them. Or maybe you secrete a chemical which causes your predator to molt a little bit faster. These are all things you can use to protect or defend yourself. Okay, that was 26. 27 is flowering plants reproduction. For flowering plants reproduction, what do you need to be thinking about? That's right. Do you think you should have that thing nailed? Yes, alternation of generations. Sporophytes always make spores. Gametophytes always make gametes, right? So you just got to know where meiosis occurs. If you know where meiosis occurs, you got to be able to figure everything else out, right? So sporophytes always make spores. When they make the spores, they're going to do it through what? Meiosis. So spores are what? Haploid or diploid? Haploid. They can be homosporous or heterosporous. If they are homosporous, they will make one type of spore, which will grow up into one type of gametophyte. If you are heterosporous, you will make two types of spores and two types of gametophytes. If I'm a megaspore, I'm probably a female. If I'm a microspore, I am a male. Same as sperm and egg, right? Sperm are smaller than egg. Same idea. Keep the pattern. All right? Now, a gametophyte, is it haploid or diploid? What's a gametophyte, haploid or diploid? Haploid. Where did it come from? A spore. Who was? Haploid. Gametophytes always make gametes, which are what? Haploid. Two haploids get together through the process of fertilization and form a diploid zygote. That diploid zygote now will grow up through what process? Mitosis into a what? Sporophyte. Sporophytes always make spores through what process? Meiosis. Those spores haploid or diploid? haploid. They do mitosis to make a gametophyte. The gametophyte does mitosis to make a gamete. Gametes get together through fertilization and form a diploid zygote, who does what? Mitosis to make what? A sporophyte. Every step has mitosis to the next one except sporophytes making spores. Okay? Sporophytes open the door to make some spores. I don't know. Think of something. Okay? Think of something. I don't know what that is. Now, let's think about, we reviewed basically 50 billion chapters, roughly 50 billion, okay? And then now look at your essay questions, okay? So your essay question, the first one says, diagram and describe the lytic and lysogenic cycles of a bacteriophage. Do you know the difference between those? Where do they overlap? Could you find 1,000 pictures of those on the internet? Yes. Did we discuss it? Yes. Which one is quicker? Which one has would you refer to it as a prophage? Lysogenic. Which one is integrated into the host chromosome? Lysogenic. Which one lies latent? Lysogenic. Okay? So you would want to be able to do a diagram of that where I see both of them interchanging and very simple diagrams. I would have things like this, right? Okay, and then I would say, and what was the phrase we used for this? APBio, mister. Okay, attachment, and then now I've moved it, if I do this, penetration, right? And then I'm what? Biosynthesis. So now I'm going to make a bunch of these pieces, right, in here, and I would draw it so I could see all the individual pieces. I'm doing it very rapidly, but you would do a much better job, right? Then what comes after biosynthesis? Maturation. I would see inside of here in maturation, I would see a bunch of viruses all mature, right? Does this make sense? Yeah. And then on, what's this? Release, I would see bust a move, and I would be seeing several viruses exiting. Yes? Okay, now, if I'm doing a diagram that's in a circle like that, and I have A, attachment, biosynthesis, maturation, release, right, and I have these pictures in a circle, how do I attach on the lysogenic cycle?
Well, after penetration, you go and do integration?
Yes. So I have attachment, and then I have penetration. I would see the DNA, and then I'm going to come out here, and I'm going to see a chromosome that has the virus in it, right? And then I can bring it back in when what? What stage? Biosynthesis, right? And I could bring it back in, and I would be writing lytic cycle over here, and in this circle I'd be writing lysogenic cycle. We all right on that? Okay, and I would make my diagrams clear. Don't give me any picture where it just is like, I don't know what that is, okay? I ought to be able to see things in there. If that's something I might do, is that something you should practice? Yeah. Okay, so that's number one. Number two, explain the physiological mechanisms involved in conducting both water and nutrients through the plant tissues in order that they can maintain homeostasis. So if you're going to talk to me about xylem and phloem function, do you think it would be important to tell me about xylem and phloem first?
Yeah.
Xylem are what? Either what? Tracheids or vessels work, and they're tapered ends if they're tracheids, more like pipes if they're vessels. And then you would say they could be dead at maturity. And I would talk about the TATC theory. I would for sure talk about the TATC theory. And I would know T stands for what? What's the big one? Transpiration. And I would be able to define it. Evaporation of water out an open stomata. I would talk about the A. What is the A? Adhesion between what? Between the xylem walls and water. Exactly right. So water is a polar molecule, and it can attach to another polar molecule of the xylem wall. I would know TAT. What's the T?
Transpiration.
What? Tension. Second T is tension. Water going from a higher water potential to a lower water potential throughout the plant. Soil is higher water potential, root is lower, but root is higher than the xylem. And the xylem has a higher water potential than the leaf, right? So you're following it all the way through, up and out, going from a higher water potential to a lower water potential. That is tension. What's the C? Cohesion. What is that?
Water.
Yes, because of those hydrogen bonds between them. And that holds them all together so that they can move together as a unit, as a continual unit up through the plant. Okay, if I'm going to talk about phloem function, that is the pressure flow hypothesis, right? Whereas TATC is about the pull, this is about the push. So first I'd want to know, what about phloem? Well, phloem is made out of what? Sieve tubes and companion cells. Sieve tubes do not have a nucleus, but the companion cell does and possibly controls it. They must be alive at maturity because you're pumping sugars, right? And water's following, which is increasing turgor pressure, which is pushing it from the source to the sink. Source to the sink. Okay? So in that situation, you're using, again, a proton pump and letting hydrogen escort sugar to another cell, which does what? When it moves the sugar to another cell, what does it do to the water potential? Lowers it. So who wants to come into it? Water. Okay? So it ends up raising the water potential, but that increases the turgor pressure. It can't go back to where it came from. That's the source. The pressure's already great there. It's got to go to where there's less. So it goes from a higher to a lower. Again, you just keep following the sugar, pushing it into the next cell. Okay, that's the pressure flow hypothesis, and that is number 2. Number 3, concerning plant reproduction and homeostasis, diagram and label, alternation of generations. Did we just talk about it? Mm-hmm. Okay, I'm not looking for details here on this one. You know, a tree is a sporophyte, simple tree. Okay, as well as the parts of a complete flower. All I want to know is like stigma, what? Style, ovary, that's called a pistil. Female picks up the pistil and says stamen, and an anther and a filament and some petals. That's good. No peduncle. You don't have to worry about that. Now, I just want to know the important parts. Identify and explain various ways that plants maintain homeostasis through feedback mechanisms. And what do I want you to think about there? Hormones. Okay? And you could, depending, I'm probably going to give you a little bit more detailed question. I'll break it down into, that's a general question, but I could talk about hormones, I could talk about tropisms, I could talk about flowering. All of those things are helping it to maintain homeostasis, and those we already reviewed. Okay, glad you came. Questions? Yes?
I didn't get on the second essay. And they're asking, when you're talking about water always follows the sugar to the cells.
Wait, say that again?
Water always follows the sugar to the
Yes,
cells.
yes.
How is it decided where the sugar goes in the first place?
Okay, that's when you want to talk about source and sink. So if you are a plant that's generating a lot of sugars, then you're going to be able to pump those sugars to a neighboring cell, active transport, right? Because you're really pumping the hydrogen and then you're getting your way in, using the water to escort it in. Ghost child. Okay, so so if you have a lot of leaves on your trees, your leaves are going to be your source. Your sink is going to be your roots and your growing part. But if it's winter, now the leaves aren't there anymore, so your source is in the what? Roots, and the rest of the plant is your sink. It's always going to go from where the sugar is more concentrated to where it's less concentrated. But yes?
With the blast lab, would that count as part of Unit 5, like for the test? Because when we have to study the labs?
Oh, I didn't ask a specific question on that in here, so I'm going to say no. Count on it for your final, but I'm going to say no. But could the transpiration lab count? Yeah. I mean, I could shadow the blast lab in, but that was really something we discussed last unit, last semester. I just didn't want to overwhelm you as you were approaching your finals. And I didn't want you to worry about it over winter break, so that's why I postponed the doing of it. Good question. What else? Yes?
in the lysogenic cycle, could you explain integration one more time?
Yeah, in integration, what happens is, after penetration, so now you have that nucleic acid in the cell, it's going to have the enzymes already prepackaged in it that allow it to insert itself into the host chromosome. And if it's a retrovirus, what is it doing? It's RNA, and it has the enzyme what? Reverse transcriptase to make a DNA copy, and there's another enzyme, replicase, which makes it double-stranded, and integrase, which then inserts it into the host chromosome, and it can lay latent. Okay, what else? Now, wouldn't it be awesome if before Sunday afternoon, that by 3 o'clock on Sunday afternoon, tell me how awesome this would not be, that you have all your essays written now. Wouldn't that be awesome? Wouldn't that be something to shoot for? If you're gonna do that and you know what your weekend schedule is, you gotta start sometime, right? The hardest part is getting started. Just write one. You know, start with one. Start with the easiest one. You know, get it done. Or eat your frog. Start with the hardest one. Remember, eat your frog? You know, you have to eat a frog. Better to eat it early than let it sit in its crap all day and then eat it, right? Eat your frog. Get something hard. Done. Okay, questions? All right, you guys, have great weekends. You know I love you.
Oh, yeah.